Hello, ladies and gentlemen. My name is Flagg Flanagan. I'm CEO and chairman of DiscGenics. Very happy to be here today to talk about our 78-week data. DiscGenics is a clinical stage biopharma company based in Salt Lake City. We believe that we're realizing our vision to create a cell therapy for disc degeneration that transforms patients' lives. Our phase 1/2 interim study results show high-dose IDCT is well tolerated, increases disc volume, and produces rapid, durable, statistically significant, and clinically meaningful improvements in pain, function, and quality of life. Disc degeneration is a common cause of debilitating low back pain, as you can see at L5S1 of a degenerating disc compared to a normal disc at L4-5. And you can see this chronic and progressive condition at L5S1 characterized by inflammation and breakdown of tissue within the intervertebral disc, resulting in pain and disability. We always believe that an ideal treatment for disc degeneration would be to reduce pain by modulating the local environment here. And we believe that not only slowing, but possibly reversing the degenerative progression of the disease. And you can see it's a pervasive issue in our society, affecting more than 250 million individuals worldwide every year, leading cause of disability, $100 billion spend, primary reason for non-cancer opioid prescriptions. Again, the Ravindra paper reinforces this with even greater incidence of lumbar spine disease around the world, with a very large global market opportunity. Product description of IDCT, injectable discogenic cell therapy, generic name Rebinuputimcel, is a mixture of live discogenic progenitor cells in a viscous carrier. These cells are derived from donated adult human intervertebral disc tissue, which is allogeneic, intended for adults experiencing pain of early to moderate lumbar disc degeneration. IDCT is injected into the center of the intervertebral disc using fluoroscopic guidance in an outpatient procedure, manufactured and cryopreserved here in Salt Lake City, and will be distributed worldwide through a cold chain logistics. Company history, you can see our progress from translational science, preclinical studies, to our allowance for our IND in the US, followed by the CTN allowance in Japan with a fast track designation. Last patient enrollment of our US study in March of 2020. Last patient enrolled in Japan, April of 2021, with the build out of our manufacturing facility completion with US interim analysis and positive US interim clinical results for high-dose IDCT in 2022 this year. We work with the CBER branch of the FDA following a biologic license application pathway. In Japan with the PMDA following a pathway for regenerative medical products. And if you look at the characterization of our cell, these are intervertebral disc cells that are converted into discogenic cells over a 6 to 8 week process that comes in 2 steps, which allows us to continue to accumulate donor lots. And from these donor lots, per donor lot, we can produce between 2,500 and 4,000 doses. This is a stable non-tumor phenotype with mode of action of anti-inflammatory and regenerative effect. You can see these intermediate cells, cluster of discogenic cells, confocal photomicrograph showing the secretion of aggrecan and collagen 2, and then a flow cytometry graph showing the homogeneity of the cell population by donor lots. We've done significant preclinical data around safety and regenerative effects, 13 independent studies. The GLP studies did not identify any toxicity or adverse immunogenic response after delivery of human cells in multiple animal species, and we saw formation of disc-like tissue in multiple models, the subcutaneous pouch of a nude mice here, and then injury of intervertebral disc in rabbits, pigs, and dogs. And you can see from the histology of this rabbit study we did, a healthy disc, the sham and vehicle, and then the introduction and injection of IDCT with the restoration of tissue in this rabbit study. As we look to the ongoing clinical evaluation of our US human and Japanese human trials, In the US phase 1-2 prospective randomized double-blind multi-center study to evaluate safety and efficacy of IDCT in subjects with single level symptomatic lumbar degeneration. These were randomized in 4 treatment groups: low dose 3 million cells, high dose 9 million cells, a vehicle control, and a saline placebo. In Japan, there were 3 treatment groups of low dose, high dose, and a sham control. Each subject received a single interdiscal injection of 1 millimeter of their assigned treatment into a single symptomatic lumbar intervertebral disc. They were assessed at 4, 12, 26, 52, 78, and 104 weeks following treatment for various outcome measures. And in the US study, it was a total N of 60 with our 1 year and 78-week interim data analysis complete, which I'm going to talk about today, and our final 104-week analysis underway. Japan study with a patient population of 38, final 1 year data analysis in process, and follow-up data from both studies will be completed and in Q4 of 2022. From a safety perspective, independent data monitoring committee reviewed all data. All IDMC reviews resulted in the continuation of the studies without modification. And if you look at our efficacy evaluation, first around safety was adverse events, serious adverse events observed from day 1 to 104. Primary efficacy was change in baseline of greater than 30% at 52 weeks as measured by the 100 millimeter visual analog scale, are well known as VAS. Secondary efficacy was change in baseline at various intervals from 0 to 104, again, VAS measurement, and then also disability measured by the Oswestry Disability Index, well-known indicator called ODI, exploratory change from baseline at various intervals around quality of life using EQ5D, and then MRI-based evaluation around disc volume. We also did additional analysis, including minimum clinically important difference, or MCID, which is a clinically meaningful threshold for change in patient-reported outcomes following the intervention. In safety, I think this goes into serious adverse events and adverse events, but the most important piece here is that there were no treatment emergent serious adverse events experienced by subjects in either high dose or low dose IDC treatment groups. Very, very good safety profile, which we think bodes well for our conversation with the regulators. And as we look at minimum clinically important difference, MCIDs are clinically meaningful thresholds for changes in patient-reported outcomes following an intervention. And in VAS, we showed a reduction of 20 millimeters, ODI reduction of 15 points, EQ5D an increase of 0.08. And each of these thresholds were derived using an anchor-based method to establish meaningful within patient change. So at 78 weeks, efficacy results around our VAS mean percent change from baseline. As you can see, high dose arm asserted itself with a dramatic decrease in pain of greater than 53%, going out to 52 weeks at 62, a little bit greater than 62%. And at 78, remaining in the 60% range. We did show statistical significance at all of the time points with high dose, with placebo and low dose, showing statistics at 26 weeks, and vehicle, albeit a little bit lower, did show significance at 52. As we looked at this further, we did look at MCID using a sensitivity analysis to impute the data for dropout subjects, and only the high dose IDCT group showed statistically significantly greater output compared to MCID at 52 weeks and 78. And I'll show you this graphically in the mean change from baseline of the 100 millimeter VAS at 52 weeks with the 95% confidence intervals relative to MCID. And again, you can see that high dose was statistically significant, outpacing the other 3 arms that were not. 78 weeks, the same, high dose showing statistical significance there. If you look at ODI, you can see a significant reduction in disability at 12 weeks, which continued on out to 78. And you can see that the high dose very much separated itself from the other 3 arms with the statistical significance. And if you look at this 95% confidence interval relative to MCID, you can again see where the high dose asserted itself, and very excited about this reduction in disability, further followed by the increase in quality of life. Again, with the reduction in pain of the high dose product with VAS, you can see this dramatic increase in quality of life at 12 weeks with durability and statistical significance on out to 78. Again, at the 95% confidence intervals, the high dose tends to assert itself very nicely, being statistically significant relative to the other three arms. And in the combination of a combined graph showing all three of the patient-reported outcomes, you can see that there's dramatic pain reduction along with dramatic reduction in ODI, with a dramatic increase in quality of life at week 12. And this extends itself out to 78 weeks, showing some really good durability, which we're very excited about. In regard to MRI disc volume, we use this validated method of an automated MRI measurement of disc volume by visit of completed cases. And at 12 weeks, you see 100 cubic millimeters of disc height in the high dose, which goes out to 250, showing statistical significance in week 52. It drops off a little bit at week 78. But again, you can see that the high dose really accentuates itself over the other three arms. And while some of them do get over baseline at week 78, all the other three arms are well below baseline. And this really has never been seen before in a study that we know of a dramatic increase in disc volume. And we're very excited about what the high dose portends in terms of the efficacy of our product going forward. This is our 78-week data. We are crunching the numbers on our 104. I want to give you a snippet of that data here. And as you can see, at 104 weeks for MRI disc volume, you can see that we took another sizable jump in disc volume increase over the other three arms. And you can see that we think this is very dramatic and evident of a very good dose response for the high dose. Very excited about where we are. And in conclusion, the primary safety and efficacy endpoints of the study were achieved, indicating that high dose IDCT appears to be a safe and effective treatment for lumbar degenerative disc disease. There were no treatment emergent serious adverse events experienced by subjects in either the high dose or low dose IDCT treatment groups. By 12 weeks post-injection, high dose IDCT produced consistent, durable, statistically significant, and clinically meaningful improvements in key outcome measures of pain, function, and quality of life as measured by VAS, ODI, and EQ-5D. And these improvements were sustained out to 78 weeks. Disc volume in the high dose IDCT group increased from baseline and reached its maximum value at 104 weeks, while no observable increases were identified in the other three treatment groups. And we find it interesting that treatment with a saline placebo did not result in consistent, durable, statistically significant, or clinically meaningful outcomes throughout the study. And although there were clear numerical difference in treatment outcomes, the study was not powered to demonstrate between group statistical significant, a la being a phase 1/2. We're very excited about where we are today in our results of this phase 1/2 clinical trial. We look forward to giving you more data as it becomes available. We're very excited about the durability and efficacy of this product to help literally millions of people around the world with degenerative disc disease. I want to thank all of my fellow employees who are working so hard here every day in Salt Lake City to achieve our mission with visions and values that are incomparable in terms of what they do. Thank you for that. And to everyone, be glad to answer any questions at your convenience.